Thanks for coming today. Today we have Dr. Levi Beverly here with us today to talk to you about neurodegeneration and cancer and the link between the two. I'll leave it over to you.
Great, thanks. All right, thanks for having me down, guys. Thanks for coming. I appreciate it. So my name is Levi Beverly. I'm an associate professor in the Department of Medicine. I'm also the section chief for research in the Division of Medical Oncology. In the Department of Medicine, in the Division of Medical Oncology, we have about 21 PhDs that do basic science research, translational research. And then I have some other titles, and I'll touch on the COSAIR Pediatric Oncology Research Program as well. Basically, I'm a basic molecular biologist at heart. I love to understand the nuts and bolts of how normal cells become cancer cells and how we can use that information to come up with new experimental therapeutics. So I'm going to tell you a few stories today from the lab. This seminar actually I'm giving is fairly new. I did give it to the Department of Medicine recently, but I'm just going to kind of tell you some interesting facts that I found from the literature and then kind of tell you how some of our research in the lab has kind of revolved around this idea of cancer and neurodegeneration and how they may be linked in some way. So before I get started, myself along with Dr. Leah Siskin, we run a research group. The two of us have our own independent labs. We kind of run it as one, and we call it the Tumor Genesis and Therapeutic Outcomes Laboratory. And we bring lots of different expertises, and we collaborate with lots of people to kind of look at cancer and neurodegeneration as larger entities, not just focus on the cancer, but also think about off-target toxicities of the cancer therapeutics, and try to come up with new models. We've done some pig work, and we've done some other kinds of development of models to address cancer as a whole and other diseases. Okay, so we're going to define some things as we go through the lecture today. So everybody knows what cancer is. I'm sure you know what neurodegeneration is, but just so we're all on the same page, cancer is defined as a group of 100 diseases or so that occur when normal cells in the body divide uncontrollably and spread to other parts of your body. So essentially, your cells gone rogue and uncontrolled growth. The flip side of that is neurodegeneration, where it's a gradual loss of the structure or certain cells within the brain or the nervous system that then lead to a disease. And so if you kind of think about this, set back that all of these diseases, most diseases are a balance of new cells being developed, old cells dying, or cells that don't necessarily divide, not dying when they're not supposed to, okay? Whereas cancer, you get too much cell growth, neurodegeneration, you get too much cell death, okay? So that's a very simplistic way to think about that. And there are a whole other host of diseases that can fall within these categories of an imbalance between the homeostasis of life and death. And so if you just think about it in one way, the cancer and neurodegeneration should be two ends of the spectrum. Okay, so then just to give you some more facts, there are over 2 million people will be diagnosed with cancer, and 600,000 people will die this year in the United States. There are approximately 20 million cancer survivors living right now, and by the year 2026, that number is going to jump even more. So there's 7 million people that are currently living with the neurodegenerative disorders, with Alzheimer's being the most people living with. So I was doing research for many years, and these weird kind of ideas kept popping up. And I finally dove down a rabbit hole one day, and I was thinking to myself, is there any relationship or inverse relationship between cancer and neurodegeneration? And so I'm going to show you some of the literature. It's a lot of epidemiological literature, and a lot of it can't really be proven or disproven. It's just observation of what happens. And so some of the first things that some of these epidemiological studies were able to show, so this is a paper from Jane Driver in 2012. And basically what this paper showed is that cancer survivors have a lower risk of developing Alzheimer's disease, and Alzheimer's disease patients have a lower risk of developing cancer. And this is once you kind of normalize across everything, age, race, smoking, whatever it is. And so that's kind of an interesting thing. So the next question is, so everything is adjusted. And so this is not just one study. There's been multiple groups and multiple papers have come out. And this says that the risk of cancer patients with Alzheimer's dementia is halved. And the risk of Alzheimer's dementia in patients with cancer was reduced by 35%. So again, two kind of interesting inverse correlations between the development of Alzheimer's disease and cancers. And one other interesting observation that was made is that survivors of the atomic bomb in Japan, they have an increased risk of cancer, obviously, because of the atomic bomb, but they actually have a 70% lower, if they get diagnosed with Alzheimer's disease, they actually have 70% less cancer incidence. And so, that's another one of those weird, like, well, how can these two things be inversely related in this situation? And then it's just another article that said that basically rate of memory loss within people with cancer is different following cancer diagnosis. They have less memory loss. And so one question might be, well, maybe it's because cancer patients get treated with something, and maybe that treatment is directly affecting brain cells or Alzheimer's development. And so it doesn't have to do with the treatment. There's lots and lots of different treatments, and people, even the same tumors, can get treated with multiple things. And so there was this huge study that was done on the veteran cohort. So these are, mostly males, 4 million almost veterans over the age of 65. And basically what they found was that veterans who survived a variety of cancers had lower Alzheimer's risks and developed less. And cancer treatment was independently associated with lower Alzheimer's risk. However, if you look at a particular type of cancer, bladder cancer, sometimes you get systemic chemo and sometimes you get localized radiation. And what this is showing is that if they were getting systemic chemotherapy, they had a 20% lower chance of getting Alzheimer's later in life. But if they got localized radiation to the same cancer, then they had no difference in their development of Alzheimer's. So again, potentially suggesting that maybe these cytotoxic chemotherapies that are given to cancer patients actually might do something to protect in the long run from Alzheimer's disease. Again, it's hard to develop, to design studies that, because most of the time you're not gonna be able to give chemo to somebody who doesn't have cancer. Again, these are just observations that occur over and over again that kind of get us thinking that there might be something to this. And so one thing you might be thinking is that, well, Alzheimer's and cancer are completely different diseases. They're driven by different molecular drivers. They have different pathologies. They're probably not similar. Well, actually, if you look at lung cancer, just for example, which is what my lab studies most, and then you scan the literature for things that are known, molecular pathways that are known to be involved in driving cancer and driving Alzheimer's disease, what you end up coming up with is lists of proteins and lists of signaling pathways that have been shown to be altered one way or the other in cancer and in Alzheimer's disease. And so what I'm just showing you here is a list of ubiquitin, which I'm going to tell you about later, is a protein that we work on. It's known to be decreased in lung cancer. It's known to be lost in Alzheimer's. Focal adhesion signaling is known to be increased in lung cancer. It's known to be increased in Alzheimer's, and so on and so forth. So there could be some interesting links of the same drivers in two different diseases. And so then you might think, okay, if they're being driven by similar molecular alterations and similar signaling pathways, are there examples of a drug that's known to treat cancer also affecting Alzheimer's disease outcomes? And it turns out that this is just a list of all these drugs have been used in cancer. Some of these are approved for cancer therapy, some of them are experimental, but they've all been used in mouse models of Alzheimer's disease and shown a delay in progression onset of Alzheimer's disease. So just for example, an inhibitor of EGFR or Src kinases or MYC signaling, these are known targets in cancer and therapeutics, but they've also been shown in Alzheimer's models to delay progression and onset of disease. So again, another interesting kind of parallel. And so now that I've kind of given this background and I've maybe opened your eyes to some interesting connections between cancer and neurodegeneration, I'm going to walk you through three kind of small stories that kind of bring our lab's research into the kind of this interplay between cancer and neurodegeneration. So first, I'm going to tell you about something that's called epichaperones, and then our favorite protein, ubiquitin, in cancer and neurodegeneration. I'm going to tell you a story that's quite a large collaborative project where we're using novel copper-containing complexes as potential treatments for cancer and neurodegenerative disorders. And then just briefly, I'm going to tell you about this COSAIR for Kids Oncology Research Program that I'm the director of. Okay, so I was a postdoc in Memorial Sloan Kettering Cancer Center in New York City with this guy named Harold Varmus, who won the Nobel Prize in 1989 for discovering the genetic basis of cancer. And when I joined his lab, is a large lab, and you show up on your first day of being a postdoc, and he says, okay, what are you going to work on? Say, work on cancer. And he said, yes, of course you're going to work on cancer, but what project are you going to do? I said, I don't know. Give me a couple of days. And I come back a couple of days later. I had 5 projects on a piece of paper. I slid the piece of paper to him, and he read the paper. He slid it back, and he said, I guess any of those projects could work. And that was the extent of my mentoring as a postdoc. And so one of the things I started doing is I was interested in, I had multiple projects, but I was interested in BCL2 family proteins that regulate apoptosis. So I was interested in understanding how the process of cell death was regulated. And so I did a whole bunch of experiments where I was trying to compare and contrast different family members of the BCL2 family. And one of the things I did was a large immunoprecipitation followed by mass spectrometry to identify protein-protein interactions of the anti-apoptotic BCL2 protein. And so lo and behold, I found this protein ubiquitolin, which I had never heard of. And I'm kind of the person where if it's something that's understudied or never been heard of, I'll dive into literature or I'll just get interested in using it. I like to be the first person that identifies something or, assigns a function to something that's never been assigned before. And so I found this protein ubiquitolin, and it bound to one of the 6 anti-apoptotic BCL2 family members, but not the other 5. And so we did a whole workup, and then that kind of went down a whole nother path of exploration and research that it started my laboratory here when I came to the University of Louisville. And so what is ubiquitolin? Well, so as the title says, this protein ubiquitolin can regulate apoptosis, apoptosis. It can regulate ubiquitination, and it can regulate protein stability. Let's get that important. But this is what ubiquitolin looks like. So it's a family of proteins. On the top is ubiquitolin 1. This protein has on one end, it has a ubiquitin associated domain, which can bind to ubiquitin. On the other end, it has a ubiquitin-like domain, which can bind to the proteasome, so it can facilitate protein degradation. And then it has in the middle these STI domains, which are known as heat shock chaperonin domains. So they're kind of protein-protein interaction domains as well. And so once we published that first paper that I showed on the last slide, we started doing a whole bunch of biochemistry and molecular biology on BCL2 proteins and their family. And what really kind of got us excited, well, so this is, like I said, it binds the ubiquitin, it binds to a proteasome, and this is what it's thought to do in the literature, but if you look in the literature, it does lots of things. It interacts with chaperones, it regulates proteasome, it regulates trafficking, and it can regulate autophagy as well. So it does lots of different things. It's not just taking proteins to the proteasome to be degraded. But what we got really excited with when we started working with ubiquitolin is that we found that it could actually play a role in human cancer, human lung cancer, especially is what we studied, but we've also worked on a little bit in colon cancer, breast cancer, and so on. And so we published this paper some years ago that we basically show that when you lose ubiquitolin in cancer, you can drive migration and metastasis and the epithelial to mesenchymal transition, which is important in metastatic prostate. This is just a couple pieces of data from that paper. What we're showing in panel A is that 45% of human lung tumors have an alteration in the ubiquitolin gene, whether it's loss of ubiquitolin 1, loss of ubiquitolin 2, or loss of both. And importantly, when you lose ubiquitolin 1, so importantly, if you have a tumor that has loss of ubiquitolin 1 or 2, you actually have less expression of the gene, which makes sense. Interestingly, we've chosen to focus almost exclusively on ubiquitolin 1 and 2 because if you look at all the family members in primary lung cancers versus the metastatic cancers, ubiquitolin 1 and 2 are the only ones that are substantially decreased in those, whereas the other family members are not. And same with looking at overall survival. When you have loss of ubiquitolin 1 or loss of ubiquitolin 2, you have a worse outcome in the clinic. And so that's why we focus mostly on ubiquitolin 1 and 2. And so since this paper, we've published a number of papers that show lots of different things and lots of different signaling pathways. And so this is an example of a Western blot. When you knock out ubiquitolin 1, or if you knock out ubiquitolin 2 with siRNA technology, what you can see is that ubiquitolin 1 is the lower fat band, and ubiquitolin 2 is the upper band. When you knock it out, you get activation of EGFR, you get activation of Src kinase, you get activation of focal adhesion kinase, and AKT. So you might recognize some of those names from a few slides back. Interestingly, when you treat these cells with a heat shock protein 90 inhibitor, 17-AG, you block the activation of many of these signaling pathways when you knock down ubiquilin 1 or 2. So suggesting that loss of ubiquilins drive all these signaling pathways in an HSP90 dependent manner. And this is just kind of reiterating it. So if you search the literature, when you have high HSP90, you have increased migration, you have increased integrin fat, you have increased SART, increased MYC, increased EGFR. When you lose ubiquilin, like I'm showing over here on the left-hand side, you get all these phenotypes as well. So there's an inverse relationship between HSP90 expression and ubiquilin expression, such that on this right-hand side, if you block HSP90, you block all of these things here. Okay, so then it kind of led us to this very simplistic model where ubiquilin is, and a lot of data I'm not showing, ubiquilin can bind to a number of proteins in the cytosol and hold them in an inactive state. Okay, so when you lose ubiquilin, these proteins then are activated in an HSP90 dependent manner. Okay, and so this is kind of was observations that we had some time ago, and we could never kind of connect a bunch of dots. And so now I'm going to tell you about a story that we'll circle back to this HSP90 stuff with ubiquilin, but we have to define some more terms. Couple of the terms are a little bit newer. So HSP90 is called heat shock protein 90. Okay, and it's what we call a molecular chaperone. So the definition of a molecular chaperone is a protein that assists in the assembly of folded subunits into oligomeric structures, as evidenced by work done back in the 80s, or it helps proteins not to become misfolded or aggregate. So a chaperone, so a molecular chaperone is one protein that can do this, but a chaperone is a whole assembly of cofactors and chaperones that work together. And this was kind of defined back in 2006. Okay, so fast forward, another 12 years, and there was a paper that came out that defined something new, which is now called an epichaperone. So an epichaperone, okay, so a chaperone, which we defined in the last slide, it's under normal conditions, it is highly dynamic. They can assemble, they can disassemble, they can interact with client proteins, and this chaperone is known to contain at least 300 different members at any given time. This new thing that's called an epichaperone is essentially a rewiring of all of the protein-protein interactions within a cell. And this epichaperone is a long-lived, stable complex of proteins within the cell that cannot easily disassemble and reassemble. Okay, and so it's just kind of shown here on the bottom is that you have your chaperones. They sense stress, there's heat stress, there's protein stress. They cause these little chaperones. They come and they go. But once you have a chronic stress, like maybe cancer or neurodegeneration or things that drive those processes, now all of a sudden you get rewiring of the protein-protein interactions within the cell, and you get this epichaperone. Okay, so this was the paper in 2016 that defined epichaperones for the first time. This is from a lab of Gabrielle Kiosis and at Memorial Sloan Kettering as well. They were interested in studying HSP90 itself and understanding what HSP90 interacts with in the cell and what regulates those interactions, more or less. I'm not going to go through all the details, but what they started out doing is something called isoelectric focusing. So it's a way to kind of identify native protein structures and where they are within once you isolate them from cells. And what they found was this. This is only part of the figure from this big paper. But what they found is that they could identify within cancer cells multiple types of HSP90 containing complexes. So some of these, well, let's start here. If you look at this column here, this is non-transformed cells. So these are non-cancer cells. And what they found using different techniques is that HSP70, HSP110, HSP90, they form small complexes within non-transformed cells. These are, and then there are some cancer cells, which are in the middle here, which they call type 2 cells, which also have smaller HSP90 containing complexes. However, there are a subset, turns out a significant subset of cancer cells, when you run these native page gels, you get this big, huge smear, suggesting that HSP90 is in a very stable, very large molecular weight complex within these cells. And so this is, and they call these type 1 cells. And so in the bottom right, this is a type 2 cell or a normal cell. You have HSP90, it interacts with different clients, and HSP90 and HSP70 are in different complexes. But when you get this state established within a cancer cell, many cancer cells, you now get this epichaperone where everything comes together and binds as one large, stable, static complex. And this is just showing, again, just to point out that there are the type 1s, type 2s, and the non-transformed cells, and they run with these different molecular weights. But the important thing here that I want to point out on the top is if you use denaturing SDS page Western blots, which basically makes every protein run at its own molecular weight, you don't really see differences in the total amounts of these proteins. What you see is differences in the amount of epichaperones within these cancer cells. And that's going to be important because HSP90 inhibitors have been used in clinical trials for many, many years without much success. And because everybody uses the biomarker was how much HSP90 is in the tumor, and then they would treat the cancers with an HSP90 inhibitor. And essentially what this is showing is that it has nothing to do with the total amount of HSP90 in the cell. It has to do with the total amount of epichaperone within the cancer cell. And so kind of for the first time, this was an explanation why some cancer patients respond to an HSP90 inhibitor and some don't. It's not about the total amount of HSP90. It's about what complex and who the protein-protein interactors are with HSP90. And so this group and others have done a lot of different work studying the epichaperone and finding drugs that are specific to the epichaperone, finding out what protein-protein interactors are within the epichaperone. And again, I'm not going to go through these papers just to kind of point them out, but they've done huge unbiased studies. They've identified hundreds of proteins that are involved in these epichaperones. They've identified some chemical inhibitors and molecular probes, and they can use these in imaging studies as well to find out which cancers have epichaperones and which cancers then might respond to an HSP90 inhibitor. This is not, it's not specific for solid cancers. It's been found, and this is just by another group as well, so it's not just all by one group. It's been found in acute myeloid leukemias as well, being important, these epichaperones. And so the question then is, so why would an HSP90 inhibitor be effective in a cell that has epichaperones and not in a cell that has chaperones? And so the idea is that, again, these small chaperones that are found in normal cells are very dynamic. They can fall apart and they can reassemble. And so if you treat those cells with an HSP90 inhibitor, HSP90s gets inhibited, that complex falls apart, but then HSP70 probably can jump back in and kind of do its job. Or HSP90 like loses the inhibitor and now can come back and do its job. But on the far right here, because you have this entire rewiring of the protein-protein interactome within the cell, now this example is very vulnerable to inhibition by an HSP90 inhibitor. And so the whole, like what they're saying here is this chaperone does a couple functions, this chaperone does a couple functions, this one does a couple. And so you can block any one of those and it might not be catastrophic to the cell. But now when you rewire the protein-protein interactome, now that thing is doing all the functions, and that's why it's extremely sensitive to collapse following an HSP90 inhibitor. So they found, I think it's up to 75% of cancer cells have epichaperones. They're sensitized to HSP90 inhibitors. And one important point that I didn't go into yet is that the oncogene MYC, which is one of the most famous cancer-driving genes, they've shown, it's been shown that MYC expression can actually drive epichaperone formation within cancer cells. And the reason I tell you that, so what about Alzheimer's? We've been talking about this interplay between cancer and Alzheimer's. And so essentially that this group and others have also shown that epichaperones are present in Alzheimer's disease. So again, here's a Nature Communication paper. What I'm showing you here on the right-hand side is hippocampal lysates from an Alzheimer's disease patient versus a non-Alzheimer's, non-demented patient. And what you see is in this native gel, you see these huge complexes of HSP90, HSP70, this protein hop, so on and so forth, but you don't see differences in total HSP90 or HSP70, again, similar to the cancer story. So again, these Alzheimer's disease brains have rearranged protein-protein interactions, which look to be full of epichaperones. So why was this intriguing to us? I already told you that ubiquitine loss drives all these signaling pathways in an HSP90 dependent manner. So I already told you that. So, okay. So, and what I didn't tell you is that we published a paper recently that said when you lose ubiquitine, you drive the oncogene MYC expression and you stabilize the oncogene MYC. And this is just showing, ignore the lower left. But when you knock out ubiquitine 1 or 2, you get a huge increase in MYC, but not when you knock out ubiquitine 3 or 4. And there's 2 different cell lines here. So MYC protein levels are increased and the protein is stabilized. And I already told you that MYC expression is known to drive epichaperone formation. So then the question is, does ubiquitine regulate epichaperone formation? So these are those nasty native page gels that you have to do to show these epichaperone complexes. What I'm showing you here on the far left, this is native page, just a Western blot for ubiquitine in 2 overexpressing cells. So you see increased ubiquitine just to prove that we're overexpressing the gene. And then on the right-hand side, what we're looking at is HSP90, HSP70, and heat shock factor in these epichaperones. When you overexpress ubiquitine, you decrease epichaperone formation, which is, I've been showing you everything with knockdown of ubiquitine, driving epichaperone probably. Now I'm just showing you the opposite, that if you overexpress ubiquitine, you can decrease epichaperones. And then this is the experiment where when you knock out ubiquitine, you can increase epichaperone. And so this is, again, native page for HSP70. So this O and M are knockout of ubiquitine. O and M are knockout of ubiquitine. And you can see it's in multiple HSP90, HSP70. And so basically what I'm just showing you is that there's an inverse correlation between ubiquitine and epichaperone formation. And these are lung cancer cells that we've done. And so we started to kind of hopefully start to connect some dots where ubiquitine 1 and 2 can, when they're lost, it drives tumorigenesis. And we know it activates all these signaling pathways. All those signaling pathways are activated in a HSP90 dependent manner. Other groups have shown that epichaperones can drive cancer and they can drive neurodegeneration and they can be potential therapeutics. What I briefly mentioned earlier is that ubiquitine 1 and 2, when they're lost, they drive Alzheimer's disease and ALS, amyotrophic lateral sclerosis. In fact, ubiquitine 2, mutations in ubiquitine 2 can actually cause early onset juvenile ALS in patients. But I will point out that ubiquitine does drive other phenotypes and other signals that are not HSP90. So it's not like everything that ubiquitins are doing are dependent on HSP function or epichaperones likely. And a big question here that we need to answer still is, can we treat ubiquitin deficient tumors with an HSP90 inhibitor in vivo and see tumor efficacy? So that's kind of that connection between ubiquitin cancer and and neurodegeneration. And so now I'm going to tell you another story that we got going in the lab that's bridging neurodegeneration and cancer. And so this is a project where we're collaborating with some folks in the chemistry department, and I'll show you them in a second. It is novel copper complexes as potential cancer and ALS therapeutics. And so this is the team that is working on this paper, Doctors Buchanan and Graberhouse, myself, and some people who were previously on the project. Haishun Guo is still on the team. Paula retired. Andrew, Sarah and Kritika moved on to other things. And so this is a very large, very multidisciplinary team. We actually meet the 4 PIs over here. We meet every Friday at 8 a.m. to discuss because there's, as you'll see, there's a lot of chemistry involved. There's a lot of radio imaging involved. There's a lot of mouse models and cell culture. And it's really not something that one lab is likely to be able to do all by themselves. And so in order to understand where this project came from, I have to kind of just describe one particular compound that's called copper ATSM. So copper ATSM is a small planar molecule that can harbor one copper compound, one copper molecule. And the reason that I'm mentioning this is because this has been around for some time and it's been used or tested, I should say, as potential imaging agent as potential cancer therapy, and it's been in clinical trials for ALS patients. But unfortunately, it did not have a lot of success and it had some toxicities. But the idea is that these small molecules can get into cells and they can either do a couple things. They can either, the copper can be reduced depending on the environment within the cell. So maybe in a cancer cell, the copper gets reduced to copper 1 and then causes copper toxicity. Whereas in a normal cell, such as ALS or Alzheimer's, they're known to have an imbalance in copper within the cells. And so you could be delivering normal copper to those cells in order to help protect the cells from the copper imbalance. So this molecule has been around for a while, and at some point down the road, or back in time, Craig and Bob decided that they were going to make a bunch of variations on this particular molecule, maybe as an antibiotic or other potential uses that have been reported in the literature. And so the chemistry folks did chemistry things, and they made lots of different molecules that had different side chains and chelated copper in different ways, and all those things. Again, the chemistry that's way beyond what I know anything about. But it's important here to point out that this library of molecules includes copper ATSM as one of them. But when they made all of these other compounds, they gave them to the lab. At that time, I think it was Paula and Sarah in the lab. We've done some of these as well recently. But tested them against cancer cells and then tested them alternatively against a non-cancer cell. And so what I'm showing here is that copper ATSM, which is number 6, you basically have a 4-fold selectivity to kill the cancer cell versus the non-cancer cell. But within these libraries of compounds that were made, you can see that some of these have an increase up to 20, 24, and 50-fold selectivity. So we've increased the ability to kill cancer cells, whereas we've decreased the ability to kill normal cells in a cell culture dish. And so this stuff was published a couple years back, just describing many of these complexes and describing some of the initial cellular biology assays. And so we've done a lot of other things with these compounds. We've done some cellular uptake assays, again, with Haisheng Guo. You can label these with copper 64, and we can do uptake assays in vitro. And one thing I just want to point out is 2 of these compounds here, they have much higher ability to be taken up into the cells. As so you see, the y-axes are much higher percentage of the incubated dose gets into cells. We've also done some imaging experiments. So again, this is copper 64 labeled molecules. We always compare everything to copper ATSM because we're trying to find something that might work similarly but have less toxicity and more efficacy. On the bottom, we're just showing here, these are mice that have tumors that express luciferase. When we inject these mice with copper 64 labeled compounds, you can see here on the arrows that some of the compounds do get into the tumors. A lot of it goes into the intestines and the liver. And so these data, with a bunch of other that I didn't show you, led to a couple many grant submissions. First chance, we almost got funded and then it didn't. And then we resubmitted the grant and it didn't even get discussed. So we kind of had to go back to the drawing board. We submitted a new version in June. But in the meantime, and this was just before Dr. Bates retired, she decided that maybe why don't we try another indication? Why don't we just see what else we can maybe use these compounds for? And so it turned out that there are a number of papers in the literature, kind of I already discussed, is that copper ATSM itself has been shown to delay the onset of ALS in mouse models, SOD1 mutant mouse models, which is one of the most common models that people use. I'm not going to go through the data, but there are multiple publications that show treatment of mice with copper ATSM can slightly delay the onset of this neuromuscular disorder. And so we wrote a grant that we submitted to the Department of Defense because they have ALS research, where we're going to treat ALS mice with our compounds. We're going to do some biodistribution into the brain. We're going to treat some neuro cells in culture. And we ended up getting funded to do this work. And so all of a sudden, I hadn't really done any neurodegeneration work, and I hadn't used any mouse models of neurodegeneration previously, but now we're right in the middle of treating a large cohort of ALS mice. in the lab with our copper compounds. And so the idea is that we hope that you can use these copper compounds as maybe diagnostic for PET imaging for cancers and neurodegeneration. If the compounds can be taken up into cancer, then we can give a therapeutic dose that can hopefully delay the cancer or help prolong life for the patient. And alternatively, we could use our labeled compounds to see if they can get into the brain, and if they can, this would be patients that might benefit when they have early onset or they have early signs of ALS. It's possible that we could use these compounds to treat the patient. And so again, so hopefully, one indication could be used for 2 different diseases. I mean, it's still early days, but we're hopeful that we can do that. And then lastly, this is just kind of a plug. It's more for, I think, basic researchers over on the other campus, but just so everybody knows, I'm the director of the COSAIR for Kids Pediatric Oncology Research Program. This was a program that was established many years ago from a gift from the COSAIR charities. The mission of the program is to facilitate and promote research in pediatric cancers and develop a robust drug discovery pipeline. It supports many investigators over at HSC, many publications and patents that have come from this program, funding from this program. Dr. John Trent was the director until he retired, and then I kind of took over for him. And so what do I do in the capacity of the program? Well, so we fund a lot of different projects that mostly are aimed at identifying new targets or developing drugs against new targets that might be involved in pediatric cancers. This is just a list of some of the people who have been funded and what their projects involve. And so what I'm doing as director of the program is continuing to support many of the projects that have been funded by the COSAIR to make sure that they continue to make progress. We've established a small laboratory that we've outfitted with a lot of different pieces of equipment that'll be helpful in progressing pediatric cancer research over in the CTRB. And we have a small, I call it a core, but it's really just one technician who does experiments for anybody who is interested in doing experiments in pediatric indications. So within the core, we have many pediatric cancer cell lines, so leukemias, lymphomas, brain cancers, bone cancers. We have lots of kind of reagents that can be used. And so the whole idea is that if you're a cancer researcher and you work on lung cancer, you don't have the reagents or the expertise or the ability, maybe because you don't have enough manpower, to then try to test your particular therapeutic in a pediatric indication. And so they would come to us, and we've done this with many dozens of different compounds that within the cancer center. We've said, okay, you've got an indication that looks like it works really well for lung cancer. Let's try it on pediatric leukemia samples. And we've done that. And we actually have one that we're about to test in preclinical for treatment of acute myeloid leukemia in pediatric patients, because we found one drug that worked amazing in these AML cells within cell culture. And we had lots of things that maybe worked on in the investigator's indication, like breast cancer, and didn't do anything to the pediatric cell lines. And so it's just kind of a way to maybe find new drugs that would not have been even tested in a pediatric setting. And this is just what the COSAIR for Kids Pediatric Lab looks like. You see here on the left, we have a hypoxic workstation where you can regulate the oxygen levels by pushing nitrogen into the workstation. You can culture your cells in there and do different things to see how hypoxia alters the biology that you're looking at. One of the reasons we got this hypoxic chamber is because we started a collaboration with a guy up at Dayton Children's Hospital, and he's a pediatric brain surgeon. And whenever they do a resection on a pediatric brain patient, they take the sample and put it in the lab. Some of these brain cancers out of the patients only grow under hypoxic conditions. So it's something that people don't think of when you do tissue culture in a lab. It's 21% oxygen, but there's nowhere in your body that's 21% oxygen. In fact, in a tumor, it's probably going to be less than 5% oxygen, especially in the brain. So you're putting these cells out of a body into a completely different environment, and that might not be physiologically relevant at all. We have a couple other pieces of equipment that's not really important right now, but if anybody's ever interested in pediatric indications, you can come talk to me. A lot of people thank for all this work and within the Total Lab, past members. Michael Scherzer was actually a master's student in bioengineering in my lab. He got his master's degree. and he just got his PhD from the University of Utah. And then everybody that's involved and been involved in the copper project. And I spoke way faster than I thought. And without that, I will take any questions of the funding. I'll take questions.
Any questions for Dr. Beverly?
Stunned. Yes.
So with that last one, delivery thing for the offer,
Yeah.
have you tried to looking at other alloys or, subsect, the delivery project with other? Like I heard gold was a possible treatment for cancer as he could like.
Yeah. Nick can tell you all about using gold nanoparticles to treat cancer. We had a huge collaborative project on that at some point back in the day. You can load it with other ions, but I haven't personally done any of that work, and we've stuck with copper just because on a lot of the assays, the copper is the most effective, at least in the cell culture setting. Yeah, so thanks. Any other questions? Yep.
Regulation of a lot of the protein pathways we also see in the cancer section with
Yeah.
loss.
Mhm.
Have you looked at the effect on like cytoskeletal reorganization or actin formation?
Yeah, so one of those first papers that we published, we looked at like actin cytoskeleton with like immunofluorescence staining, and we have looked at some other like RAC and RO, GTPases, and some things like that. We've never really, from a biomechanical or like biophysical or any of those, we haven't really gone much into depth other than Western blots and immunofluorescence. Yep.
Thanks for that great presentation. It's great to think about what you're working on and great that we have this. I have two questions.
Yeah.
First one, I don't think I've ever heard of that link before.
Yeah.
Between Alzheimer's and, cancer
Mhm.
inverse relation, which is very fascinating. Now, for chemo treatment, where you show that chemo treatment or, there's data shows that can actually reduce that risk. But one of the side effects for chemo treatment is can have neurological effects, right? So some people have
Chemo brain.
Yeah,
Yeah.
so I assume that was separated to be able to figure out what's actually Alzheimer's and what's just.
Yeah.
Neurological effects from the chemo.
Right.
Treatment. So that's my first question.
Yeah.
And then if there's time, I'll ask the second one after that.
The short answer is, I don't know for 100%. But I can say that it depended on the therapy, and not all cancers have this inverse correlation as well. So I think like melanoma, there's no correlation between inverse correlation. So it's not all of them, but like lung cancer is one. And the one example with the chemo was bladder cancer. And again, I've talked to neurologists about this and other people, there are possible other explanations. So this is one of the problems with any epidemiology stuff, right? It could be true and unrelated, unfortunately. Or maybe people with Alzheimer's disease don't know that they're sick from cancer as well. And so then they don't get the cancer diagnosis because they never get looked at, right? Or something else with the cancer patients feel terrible and they don't go get diagnosed with Alzheimer's disease later. I don't know. A lot of these are, if you look more out, like 10 years after the cancer treatment, right? So.
But you showed the data from different
Yeah.
populations, and then you also are seeing interesting trends with like.
Yeah.
I had a different question. So you start out with the ubiquitin effects, and then you identified this whole HSP90. The focus is obviously a significant role. How did you choose to look at HSP90? Was that your first thing? You just thought, well, we should look at this, or did you screen through others? Because it makes it look like you're like, let's look at HSP90, and obviously that was significant.
Right.
But
Yeah.
I think sometimes for students to learn, how does the tracing process work? Was there others that didn't show that?
Yeah, so a lot of the things that we started with were like, okay, so I'm trying to think. we just had panels of antibodies and we started doing things, right? Okay, well, let's look at EGFR activation. Let's look at AKT activation. Let's look at FAK signaling activation. And we would find those, and then we would go back and we would use a FAK activator or inhibitor, and then we use a SARC inhibitor. And so we kind of just, as we found things, we decided to try those individual inhibitors. And we're trying to kind of put things in order, like what happens and in what order when you lose ubiquitin. And it just turned out that like that one little table that I had, we had these huge lists of things that were changed when you alter ubiquitin. And then at some point we kept seeing that HSP90 can regulate that and HSP90 can regulate that and HSP90, and like all of a sudden when everything started adding up and equivalent, then we're like, okay, well, maybe we need to look at HSP90 now. So yeah, it wasn't the first thing that we looked at for sure.
A logical progression because.
And looking in the literature, I mean,
Yeah.
without making those connections within the literature,
I always find it helpful to hear how people make these discoveries because as we're doing our research,
oh, for sure.
going through this process, that's not something you will necessarily hear as you read an article or something. So,
And things are never presented in the order that they happen, right? Because that doesn't make a good story for the most part. So, yeah, when you're like, oh man, how they go from that to that, that's amazing. And I'm sure actually that was actually at the end and then they did 1,000 things in the middle, but it just makes sense to tell the story that we saw this, we did this, it worked, boom.
the results are definitely.
Yeah.
So you talked about looking at all these pathways, plan inhibitor, things of that nature. Did you ever use like a software to like model these things and track it, the software model?
So, I mean, again, the short answer, not really. Like we did some things like, it was back when we first started this microarray technology for mRNA expression, and then we did some RNA sequencing. But again, that's not at the protein level, and it's not showing you which pathways are activated necessarily. It's just giving you the gene signature downstream. So we had done some gene signature stuff. And that's actually how the MYC story that I told you about, that's how that came, that particular story came from doing the RNA sequencing of what genes are changed when you knock out ubiquitin. We fed that into a protein, an algorithm called gene set enrichment analysis, GSEA. And it basically told us what you just put into this program looks like a MYC signature. And so then from that information, we went back and looked at MYC, and lo and behold, MYC was increased and it was stabilized. And so we were able to make a whole story out of that from that piece of software. As far as the protein signaling pathways go, we have not really, we tried to do some more larger, unbiased kinds of experiments with that, but most of that was just, oh, we have an antibody, let's throw it out of Western blot and see if it works. And something happens. Sometimes it does, sometimes it doesn't. Sometimes you get lucky. Important thing about research. Sometimes you gotta get lucky.
Any other questions for Dr. Beverly? Yes.
Yeah.
Talking about a lot of this like trial, throwing things out. Just like curiosity, did you find anything like strange or interesting outside of what you just reported? Something that's like kind of crazy, like a new protein.
Yeah, I mean, there's always something. One of the problems that I had more so when I first started my own lab was we had lots of different directions. Everybody had their own little project, which in retrospect was probably not the best way to go about it. But every time I would see something, like I said, like I found a new protein that interacted with something that there were no papers on it, right? And I'm like, well, what does it do? It's got to do something. It wouldn't be there. And so then I would follow that and make a story out of it, and then it would just not really turn into anything amazing. One story that we had, we just published recently, we were looking at the microbiome within lung cancers. And so just I'll give you the minute synopsis of that. I had a graduate student who was looking at nutrient deprivation in cell culture and lung cancer cells. And I have a friend in Canada, and they had done biopsies on human lung cancers and right next to the lung cancer. And they were doing microbiome studies. And they found that the microbiome within the lung tumor was different than like a centimeter outside of the tumor. And long story short, what we found in collaboration with them, we did most of the biology work, is that microbiome within the tumor is enriched for a population of bacteria that can produce methionine. So we cannot produce methionine. It's an essential amino acid, but bacteria can produce it de novo. And it turns out that we found that it's possible, and again, this is all kind of hypothetical, but we showed that bacteria can pump methionine into the media and cancer cells can take that methionine up. So our hypothesis is that it's a symbiotic relationship within the tumor because within tumors, you have nutrient deprivation. And so now methionine, which is essential, is limiting. It selects for a population of bacteria that can secrete the methionine, which then the cancer cells take up. And then the cancer cells, actually, we showed, can give some stuff to the microbiome as well. But anyway, that was kind of an interesting, cool story that we found. And that was a kind of a happenstance collaboration that we didn't actually go seeking. It just, we bumped into them. So anyway. All right.
With that, let's give Dr. Beverly a thank you for coming today. Everyone, please don't forget to sign in using the QR codes that's been placed on your desk. Other than that, have a good weekend.